Yesterday we were discussing about the secondary lymphoid organs plus the primary lymphoid organ and had given you the detailed structure of thymus and its function as well as spleen and lymph node. Please remember, in the examination, any one of them can come for 10 marks question asking structure and function of the lymphoid organ, any one of them. So it is important that you should remember the diagrammatic sketch or the structure of any of those organs plus the functions that I have explained to you. Now we will move on to other two lymphoid organs, that is mucosal associated lymphoid organ and cutaneous lymphoid organ. When I say mucosal associated lymphoid organ, I had mentioned to you that whichever the surface that is covered by mucus or mucosoid epithelium, that region is protected by a special secondary lymphoid organ called as mucosal associated lymphoid organ, that is MALT, M-A-L-T. That is your nasal passage, the sinusoids that are there, sinuses, the oral passage, GI tract, and urinogenital tract. So just to once again recall back the tonsils and other lymphoid organs associated with our face region, we'll just see the positioning, but there is no necessity for you to remember these diagrams. It is only necessary for you to remember their position. There is a lingual tonsil. Lingual refers to the tongue tonsil, which is just behind the tongue, not in the throat region, it is behind the tongue. That is one lymphoid organ associated with your tongue. Okay. Then the another lymphoid organ which I mentioned, the pharyngeal tonsil. The pharyngeal tonsil, which is also an adenoid that is there in your throat. On either side, it is there. When it swells, it obstructs the entry of food. And you experience this enlargement of tonsil when you suffer from common cold. Then there is an adenoid here near the nasal region, just below your eyes, in the cavity here. And that adenoid is there on either side of your nasal bridge. Okay, that is also an adenoid or a tonsil. Then the lymphoid organ in a bird, which is called as bursa fabricius, which is different from the other organs. And it is this organ that was responsible for giving the B lymphocytes their name B cells, because first the lymphocytes which are responsible for the production of the antibody were discovered in bursa fabricius. Since it was discovered in bursa fabricius, those lymphocytes were called as B cells. B refers to bursa. But then when the discovery continued with higher organisms, that is human beings, they found that human beings do not have this organ, bursa fabricius, attached to the colonic region, colon or the rectal region. Okay, it is not there in human beings. Instead, the production of B lymphocytes has taken over by the bone marrow. And therefore, the scientists retained the word B, B for bursa fabricius, B for bone marrow. So the B cells take their origin from the bone marrow, whereas T cells take their origin from bone marrow but undergo maturation in the thymus. B cells, after originating from the bone marrow, undergo maturation in the bone marrow itself, and then they come out into circulation and lodge themselves in the secondary lymphoid organ that I discussed, that is the lymph node and the spleen. In abundance, they come and lodge themselves there. The next region where the B cells come and lodge themselves in abundance is the intestinal mucosa, the mucosal lining of the intestine. We will now take the discussion on the mucosal lining of the intestine. This is a highly diagrammatic presentation of the intestinal wall. This is the outside region of the intestine, and this is the lumen of the intestine where the food will be passing through. and these are intestinal villi. And the crevices or the pockets of the villi at the bottommost part of that pocket is called as intestinal crypts. CRYPTS, intestinal crypts. And these intestinal crypts are the areas where the entry of the antigens takes place in a large amount. Why? Because the food for some time remains there in these crypts for quite a long time compared to the villi surface. The food does not remain on the villi surface, it gets easily washed off and goes to the other regions, that is the colon. But in the crypt, some amount of food will be remaining, and that will be carrying large number of antigens. And the antigen entry takes place through the crypt. And I had mentioned one example in the innate immunity. The crypt area contains cells called as Paneth cells. Paneth cells are one type of specialized cells which are responsible for the production of defensins in that area. In addition to Paneth cells, there are some specialized cells which are called as M cells, mucosal cells. We'll see their function little later after I discuss this part. So the area that is very close to the villi is called as lamina propria. Lamina propria. The layer which is there, which is mucosal layer, which is very close to the villi, is called as lamina propria. The layer below that is called as submucosa, which is full of the primary lymphoid follicles as well as secondary lymphoid follicles, which we had seen in the lymph node and the spleen. Remember that these lymphoid follicles were there, where the highly congregated population of B lymphocytes and the T lymphocytes could be seen in that area, waiting for the presentation of the antigen. The submucosa area is rich with B and T cells. Muscularis mucosa, it is called as muscularis mucosa, which is the outermost layer, consists of circular and longitudinal muscles, which are responsible for contraction and expansion of the intestine. And remember, the entire lymphoid follicle area with congregated B cells and T cells is called as Peyer's patch. P-E-Y-E-R''S. Peyer's patch. Peyer's patch is the lymph node like area, but it is a lymphoid area, and there are no lymph nodes here, but these are the areas which resemble the lymph node, the Peyer's patch. Now let me take the detail of the crypt area, how it looks with the M cell. The M cell looks like this. It is a cell which is mucosal cell, modified mucosal cell, having a pocket or a cavity. In that, the B lymphocytes and the T lymphocytes as well as the macrophages can be enclosed. They will be there in that area waiting for the capturing the antigen. So the crypt area of the intestine, I said, where the food particles and the antigens, even the bacterial population is very, very heavy there. And whatever the bacteria that infects you could remain in that area for a longer duration because it is like a crevice, okay, crypt area. Then the M cells will receive the antigen, and that antigen is passed on through the M cells just like the way the macrophages do it by phagocytosis. They phagocytize the antigen. It could be the food antigen, foodborne antigen, or it could be the antigen of the bacterial origin or the viral origin. Whatever the antigen that has entered through your food or through your oral passage will be passed on to the immune cells from the M cell. M cell helps in phagocytosis of that antigen and pushes it into the cavity that is called as pocket. In the pocket, already the antigen presenting cells will be there, that is macrophage and dendritic cells will be there. Interdigitating dendritic cells and the macrophages. I had mentioned that both of these cells are professional antigen presenting cells. And as soon as they encounter the antigen, they will process it, and the macrophage undergoes activation. It will release the IL-1. IL-1 in turn activates the Th cells, and the Th cells will now form a contact with those macrophages which has processed antigen and presenting it on class 2 MHC. Through the T cell receptor, which is shown here as a brown forked structure, that is T cell receptor, it will establish the contact with the macrophage that has processed the antigen and undergoes activation. And when the T cell undergoes activation, in the vicinity, B cells also can undergo activation under the influence of macrophage by the cytokine IL-6, and the T cells also secrete cytokines for the activation of B lymphocyte. They have the receptors on their surface for receiving the antigen, and they also get activated. Now, these activated T cells and B cells can descend to this area, that is submucosa area, from the lamina propria area, and start proliferating here in the submucosa area. They undergo proliferation. Activated B cells and T cells undergo proliferation and develop germinal centers. And now they are committed for that antigen, and they are responsible for developing immune response against that antigen in the submucosa area. Let us see a little bit more detail of the mucosal immune system. So the antigen received by the M cells in the submucosa, in the lamina propria area, are conveyed to the T and B lymphocytes through the antigen presenting cells. They undergo proliferation, and they are responsible for building the immune response, where B lymphocytes will be in excess in the intestinal mucosa because the antigen entry in the intestinal mucosa is very, very high. Therefore, they have to keep on secreting a secretory antibody by transforming themselves into plasma cells, and that secretory antibody produced by those B lymphocytes is going to be poured out into the mucosal lining, and that secretory antibody is called as IgA, immunoglobulin A. And I had mentioned that immunoglobulin A has a special characteristics that it cannot be digested by our digestive enzymes because it has an inhibitory protein chain protecting it from digestive enzymes. Therefore, it can serve the defense at our intestinal surface. And the quantity of IgA produced per square centimeter area of the intestine is about 15 grams per ml. Such is the large quantity of IgA is poured into the mucous lining of our intestine, because that is the largest surface in our body. Intestine is the largest surface which is exposed to antigens continuously. Then coming to the other cells, there is intraepithelial lymphocytes which are there in between the epithelial cells of the mucosal lining. Columnar epithelial cells with villi are present on the outermost, that is then the lumen surface of the intestine, and in between them you can see the intraepithelial lymphocytes. And I had mentioned about them IEP, intraepithelial, IEL, intraepithelial lymphocyte, IEL. IEL, these cells are nothing but gamma delta T cells. Gamma delta T cells. And I had spoken about their function, that they are capable of moving in the surface of the epithelium back and forth, and whenever they come across the infection in the epithelium, that is intracellular infection in the epithelium, they target that cell and bring about killing activity of that cell. They will kill that cell. Okay, so intraepithelial lymphocytes, I had mentioned about that. And there are also other cells that are there in the submucosa area, and those are the epithelial cells which are also called as lamina propria epithelial cells. Okay, lamina propria epithelial lymphocytes. Lamina propria epithelial lymphocytes are nothing but they are T-head cells. And I told you that those T-head cells are there readily here in the pockets of the M cell, as well as they are distributed in the area, that is in the submucosa area. And as and when there is an antigen entry, they can congregate in that area and proliferate to form the follicles. So the intestinal surface is a highly defended surface of our immune system. Why? Because the entry of antigens is continuous there. There are a large number of antigens, foodborne antigens, which enter the intestine and it needs protection. And that is done by the immune cells which are present in the mucosal lining. Then comes the another area, that is our skin. The cutaneous associated lymphoid tissue, that is called CALT. Remember, I said that that is the surface of our body which is impervious to the bacteria. Bacteria cannot enter through our skin. Even though there are openings, hair follicle is opening, plus the sweat gland there is opening, but they do not enter through those passages because they are protected very well by the innate immune system. The oil and the sweat that is secreted, plus the resident population of the bacteria that is living on the surface of our dead keratin. Dead keratin means the surface area without cells, that is keratin, a protein which is lining on the surface. In that area, the bacteria, hostile bacteria, even if it comes and lodges, it is not allowed to survive. The condition is maintained like that. But still, the skin has to have its defense system. Just in case if it is punctured, wounded, what response it has to show. Let us see how it shows. Now, this is the simplified structure of the skin. The hair follicle is shown here, okay, and then you will see the sweat gland is shown here, and these are the epithelial cells. Epithelial cells are called as keratinocytes. They secrete keratin, that is the protein, and interdigitating dendritic cells that are there in the epidermal area. The uppermost area is called as epidermal area. Those interdigitating dendritic cells had named them as Langerhans cells, I had mentioned that. And those dendritic cells which are there are antigen presenting cells. By any chance, if any antigen makes its entry through the skin due to the injury to the skin, these are the cells that are going to come in contact with that antigen for the first time. Okay. In addition, I have mentioned that there are intraepithelial lymphocytes, and those are CD8 cells. I had said that these are there in the skin epithelium and the intestinal epithelium, lung epithelium. And these gamma delta T cells, which are present, which are CD8 T cells, which are also present here, they are responsible for, in fact, instantaneous killing of the intracellular pathogen which infects any of these epithelial cells. Remember that. So the gamma delta T cells, which are intraepithelial, and there are also intraepithelial CD8 T cells. And then the skin surface at the bottom is made up of dermal layer. After the epidermis, there is dermal layer. In the dermal layer, there are larger number of T lymphocytes, and those T lymphocytes are the T lymphocytes of Th cell category, and there are also B lymphocytes and macrophages. Okay, so those dendritic cells, which I had said, when they encounter the antigens in the epidermal area, Langerhans cells, which are dendritic cells, they travel from that area to the nearest lymph node, wherever the lymph node is available, through the afferent channel of the lymphatic vessel. And when those dendritic cells travel through the blood vessels, I had named them as wheeled cells. Any dendritic cell leaves a particular tissue area and starts traveling through the blood is called as whealed cell. Then it will go and lodge itself in the immediate nearby lymphoid organ or lymph node, and once again it transforms itself into interdigitating dendritic cell. By then it has already captured the antigen and it is ready to present that antigen to the B lymphocytes and the T lymphocytes, which have assembled themselves in the cortex area of the lymph node. So the cutaneous lymphoid tissue does its own function of protecting us by having the antigen presenting cells in the epidermal area as well as the dermal area, plus the immune cells that are responsible for expansion of the immune response, that is TH cells and the B cells, which are there, which function soundly and protect our skin area. And that is how the cutaneous associated lymphoid tissue functions. Okay, this is as far as the cells and organs of the lymphoid region or cells and organs of the immune system. So we will move on to the antigens. We were uh, we were talking about the antigens earlier, and several times we had made a reference for that, for the antigens. And we had also said that the antigens are of different categories, and those antigens which are of different category, those antigens which are of different category are actually belonging to the proteins, carbohydrates, lipids in complex form. Okay, and I had said that antigens will vary in different organisms, bacteria, viruses, and antigens also vary in human beings. But the antibodies produced in human beings will be always of the same class, and human beings will never produce antibody against the antibody of another person. But a human being will produce antibody against the antigens of the other person. And those antigens which are there in human beings belong to a large category, that is the HLA complex, human lymphocytic antigens, which is also called as major histocompatibility complex, MHC complex. Therefore, human beings cannot receive or transplant the organs from one individual to another individual easily. It is not going to be accepted. If it has to be accepted, the organ match has to take place, and the matching of the organs should be done up to 85%. Then remaining part of the antigen dissimilarity can be won by administering immunosuppressive drugs. We will talk about this in detail when we are going to talk about the MHC complex as such, as a separate part. For the timing, you keep in mind the human major antigens belong to MHC complex. Then, what are antigens? Antigens are substances that can stimulate an immune response. And those antigens which stimulate an immune response means they bring about activation of T cells and B cells. They bring about production of the antibodies. They are antigens. Okay, and most of the antigens are immunogenic. Immunogenic means they can stimulate the immune response. And earlier there was a definition stating that all foreign substances are antigens. No, I don't agree with that definition. The antigens are those which can induce an immune response are antigens, but all foreign substances are not antigens. For example, dust particles which enter our body, if you consider them as antigens, they cannot induce antibody production, but they can cause irritation in your body. In the immune system, they can cause irritation. They can cause problem to the macrophages because macrophages will engulf those dust particles or silica particles or carbon particles which enter through our nasal passage into the lung and make those immune cells, that is macrophages, useless. And granuloma can be formed, it can be deposited. That's all they can do, but they can't induce the production of the antibody. Therefore, all antigens cannot be immunogenic, but those antigens which are immunogenic can stimulate the immune response. That is both cell mediated and the humoral immune response, that is antibody production. But there are antigens which by themselves cannot be immunogenic, but they can become immunogenic after they enter our body, and they are called as partial antigens, which are also named as haptens, Haptens, haptens. We'll be talking about them in detail little later. Haptens are partial antigens. For example, sometimes some people develop reaction to drug, in the sense they develop allergy to some drugs. Allergic response shown against sulfa drugs, allergic response shown against penicillin and some other drugs. And those drug molecules can be called as haptens. They are partial antigens. Once the person takes these drugs for some clinical reasons, for controlling some disease, that person may develop immune response against those drugs which are administered to him, because those drugs will form some complex in some proteins that are available in his body and bring about an immune response. So the haptens are partial antigens which bring about immunogenic response. Therefore, they are called as partial antigens. Okay, but the haptens by themselves, okay, cannot stimulate the immune response. They can only form a complex and then stimulate an immune response, and those details we will deal little later. Okay, now classification of the antigens. You can classify antigens into two major categories. One, exogenous antigens that do not belong to your body. They come from outside. Exogenous antigens are those originate outside and they come from outside. They could be of bacterial origin, they could be of viral origin, they could be from the pollen grains of the plants, they can be bacterial toxins, and they can be bacterial and viral proteins. When I say bacteria and virus, it is the whole cell or whole viral particle. But it did not be always whole cell or whole viral particle. It could be the protein of the bacteria or it could be the protein of the virus. These are exogenous antigens. They are going to be recognized by our body immediately as soon as they enter our body, because we have innate mechanisms of recognition, and I had mentioned to you about the toll-like receptors that are there in the cells which are innately responsible for recognizing these antigens. And once these antigens are recognized, the immune response is going to develop against these antigens, and that immune response will be of both the types, cell mediated as well as humoral. Okay, so exogenous antigens. Then comes another category of antigens. Those are called as endogenous antigens. Those antigens that originate within our body, or those antigens which originate within the body of another animal. On the basis of this, these antigens are classified. Okay, those antigens which are there within oneself, and which is different from his own brother sister, because he is genetically different from his brother sister. There is some difference in the genetic constitution, and those antigens are called as autologous antigens. They are self antigens. Autologous antigens are self antigens. They are individual specific. The antigens that are present in me are different from the antigens that are present in you. It is only that these autologous antigens will be similar only in identical twins. Those who are born as monozygotic twins, identical twins of same sex, they will have autologous antigens similar because their genetic constitution is almost identical. But those are dizygotic twins, that is fraternal twins. They can have different sex, or they may be of the same sex, but they are not identical to each other. They look different, slightly different. And fraternal twins do not belong to this category, autologous antigens. Fraternal twins belong to allogenic antigens. Allogenic antigens. So autologous antigens are individual specific. They are like a fingerprint of a person. He will or she will have these antigens lifelong because their genetic constitution is such that they will express these antigens continuously. And one person who has this self antigens, he or she will have developed tolerance against these antigens. All his immune cells know that they are self antigens, hence they do not build up immune response against them. Only when, if the immune response builds against the self antigen, that will lead to autoimmune disease, autoimmune disorder, that we will talk in the 4th unit. But for the time being, keep in mind that it is not very common. Autoimmune disorder is not very common, and most of us are tolerant, and our immune cells are tolerant to our self antigens. They will never build immune response against the self antigens. Okay, autologous antigens. Then allogenic antigens. These are the antigens distributed across the species. Let us say human race. All human race have allogenic antigens. And these antigens belong to the category which I mentioned to you, that is major histocompatibility complex, MHC. And they are also called as HLA antigen complex, human lymphocytic antigen, HLA. The major histocompatibility complex, which determines the type of antigens that are expressed by all our tissues. Okay, our tissues will have fingerprint antigens. You can identify the liver tissue from kidney tissue because the antigens expressed by liver tissue will be different from the antigens expressed by kidney tissue because both of these tissues or organs function differently, even though they contain the same genome. They contain those 23 pairs of chromosomes, but the genes that are in action in these two tissues are different. Second, the antigens expressed by these two different tissues are different. Like that, we have different organs expressing different antigens, and all those antigens expressed by different tissues belong to class 1 MHC. They are fingerprint antigens of each tissue. Class 1 MHC of all the tissues will differ. Okay, and at any given time in a human being, okay, the class 1 MHC that is expressed by a human being, there will be 6 different types of class 1 MHCs will be expressed. They are called as 6 haplotypes. Once again, more details of this we'll be talking about in major histocompatibility complex when we are going to discuss that. But for the timing, keep in mind that MHC complex is not same, not identical. It is there across the species, and it is species specific, and it differs between brother sister, brother brother, and sister sister. And without matching this major histocompatibility complex, one cannot donate organs, one cannot transplant organs. Just like the way you match the blood groups, one has to match major histocompatibility complex. Okay, so allogenic antigens are species specific and they are individual specific. They are specific for that particular individual and they are the identity of that individual. And using this major histocompatibility complex, also one can do forensic investigation. And you can find out the relationship of the individual, relationship of the family. You can draw the family tree using MHC complex. So allogenic antigens are very important antigens and they are capable of inducing immune response when you transfer this antigen to other individual. Okay, and other individual will react to this allogenic antigens immediately and start responding to it and stimulate the immune response and build up the immune response. Then comes xenogenic antigens. They belong to two different species which are not related, xenogenic, like human being and other primates like chimpanzee, gibbon, and gorilla and orangutan. All these belong to the primate category, primate like human beings. They are tailless primates, just like they are belonging to the ape category. We belong to ape group, right? So xenogenic antigens across the species of different types, they are different and they are called as Forssman antigens. Forssman antigens, named after the scientist. I'll give an example, simple example. And I had told you that Julius Caesar used to practice punishing the criminals by transfusing the blood of animals into the human beings and kill them. Let me take an example, simple laboratory test. You separate the human serum and keep it in one test tube, and separate rabbit serum and keep it in another test tube. Now you have serum of two different species which are not related. You want to take even the apes, any apes, take the serum of apes, keep it in a test tube. And let us take, for example, human RBC. We will take human RBC and add that RBC to the test tube containing the serum of rabbit. I will add that RBC to the serum of rabbit. Same way what I will do in another test tube where I have taken human serum, to that I will add the RBC of rabbit and just see the reaction. Immediately those RBCs will be hemolyzed. That is, our serum contains inherent ability of resisting the other species blood from entry. Innate response. So that is what I am talking about. That RBCs of the human beings contain Forssman antigen, which is human specific Forssman antigen. And that Forssman antigen is going to be instantaneously recognized by the other species because of its innate response. And this is the simplest experiment one can do to prove that the Forssman antigens are potent antigens resisted by the body from entry. So you cannot allow the blood of any other animal species to enter your system. Neither other animal species can allow the blood to enter their system. It is very, very strong and very, very powerful reaction. Immediate hemolysis takes place. Immediate hemolysis takes place, which Julius Caesar did not know. By transfusing the blood of the other animal into the human beings, criminals, he was killing them because the RBCs of the human being would undergo lysis because the blood of the other animal contains the serum, and that serum would instantaneously react on our RBC and cause hemolysis of RBC. Okay, anyway, that practice is not now anymore practiced. It was in the ancient times. So it is one of the classical example of the Postman antigen. And remember, sometimes our autologous antigens, autologous, that is within us, will become strongly antigenic because they change their chemical constitution. And when they change their chemical constitution due to some abnormality, change or substitution of some amino acids or change in constitution, they behave strangely, and such antigens will become the antigens to develop immune response. And one such example is cancer. During cancer, the cancer cells will express some strange antigens on their surfaces. Therefore, cancer cells are under attack by our immune system. Large number of cancer cells die initially, but later they resist the death by developing certain mutations and resist immune response and evade immune response. This part of autologous antigens resisting the immune response or evading immune response by cancer cells will talk in unit 5. But for the time being, you keep normal autologous antigens will never stimulate immunity in a person, and person will not develop immune response against them because the normal person is tolerant to those antigens. And those antigens which are allogenic, the person will definitely resist because they are different from one individual to another individual, born in the same family. Okay, and therefore it is very essential to take note of these antigens whenever the organ transplant is going to be conducted. So I will stop here today. We will discuss other properties of the antigens and the properties that govern antigenicity in the next class. Thank you very much.